Hello, my name is Kristen Hagan. I'm a third year in biomedical engineering, and I'm an undergrad researcher in Dr. Quinton Smith's lab. For my UROP, I studied a vessel-on-a-chip microfluidic device for modeling blood vessel vasculature under diabetic conditions. Diabetes is primarily associated with dysfunction in regulating your blood glucose levels. A lot of people don't know that it actually causes severe cardiovascular dysfunction. Vascular inflammation leads to improper blood flow, irregular smooth muscle dilation, and arterial stiffness. This eventually leads to severe cardiovascular diseases, cardiovascular events, and even death. Right now, the models of human blood vasculature are two-dimensional cell culture and mouse models. And while they do work, they are not completely representative of inflamed vessels or with drug interactions. To address this gap in research technology, we developed a microfluidic blood vessel on a chip. We made a wafer using soft lithography and use this as a mold for our PDMS devices. With those, we can see the center channel with a mix of collagen and stromal cells and introduce two needles. Then when the collagen polymerizes, we can remove the two needles, and this leaves space for us to seed with endothelial cells. This makes our device representative of the three-dimensional structure, physiological conditions, and stromal cell interactions that is the same in vivo. To induce diabetic conditions, we treated the cells with pro-inflammatory cytokines, IL-6, TNF-alpha, and a high concentration of glucose. To test for these effects of our diabetic treatment, to make sure that our vessels are behaving as they would in vivo, we used PCR to check for inflammatory marker upregulation. We checked for ICAM, which is an intercellular adhesion molecule that is upregulated with inflammation, and it promotes immune cell adhesion. IL-6 is another secreted cytokine, where higher secretion indicates inflammation and oxidative stress. Col4A1 is the collagen present in endothelial basement membrane, and upregulation indicates issues with vascular permeability and stiffness. ANGPT2 is a marker of vascular instability and angiogenesis, where higher expression indicates endothelial dysfunction. All of these PCR results showed that in our diabetic conditions, these markers were upregulated compared to the controls, which means that our vessels are behaving as expected, like an in vivo diabetic vessel. In the following weeks, I will be testing for increased monocyte adhesion and imaging for feasibility in real time. I will also be administering the drug empagliflozin, which is known to reduce arterial stiffness and increase wall shear stress. I will quantify and analyze the data to demonstrate that the blood vessel on a chip mimics the behavior of a diabetic blood vessel in vivo and that it responds to drugs appropriately, meaning that it can be later used for drug development trials. Thank you.